For everyone with an interest in NASH, or more broadly, fatty liver disease, Surf's Up, Season 4, Episode 27 of Surfing the NASH Tsunami. Our second part of our preview for EASL Congress 2023, featuring abstracts of interest, starts now.
This week on Surfing the NASH Tsunami.
This EASL Congress leads us one step closer to how we're going to be managing patients with fibrotic fatty liver disease.
Can we move beyond histopathology to AI-assisted or even AI-driven reads for admission or endpoint assessment studies?
There will be so many questions still open about how we actually use these drugs in practice.
This is a screen and some evidence that liver health counts as just as much as liver disease.
Every week, a global community of fatty liver disease stakeholders comes together to explore the most important challenges in diagnosing, treating, and developing medications for patients with fatty liver diseases. Join hepatology researcher and key opinion leader Professor Jörn Schattenberg, liver wellness advocate Louise Campbell, pricing and forecasting guru Roger Green, and their guest, hepatology researcher and key opinion leader Professor Ian Rowe, as they review what they consider some of the most important and intriguing abstracts at next week's EASL Congress meeting in Vienna on Surfing the NASH Tsunami. Today's episode is sponsored by HistoIndex, the first global company to provide stain-free, fully automated imaging solutions for visualizing and quantifying morphological and architectural features of fibrosis.
HistoIndex couples breakthrough biophotonic technology with an AI-powered digital pathology system to provide consistent, high-throughput imaging in liver disease and cancer.
The platform benefits clinical research and speeds development of drugs and diagnostics.
For more information, visit www.histoindex.com.
So before we start, I should tell you this is going to be an interestingly constructed episode. Right now with me online, I've got Ian Rowe and Jörn Schattenberg. We'll say hi to them in a couple of minutes. We will talk about articles of interest to us, abstracts of interest to us. After that, I spoke with Louise yesterday. She wasn't able to be on today, about abstracts of interest to her. And then at the end of all that, we have an extra-sode sponsored by HistoIndex, talking about a trial they're doing right now, taking a look at the benefits of AI-assisted pathology reads for the purposes of drug development. So it's going to be probably an hour and a half by the time we're done. It will have a sponsor message in the middle. It'll have 3 separate conversations, which should be plenty to keep your voice occupied. And for those in the States who are going to be flying over to EASL end of this week or Monday, it will get you through at least the first hour and a half of this flight until you fall asleep if you've not listened before that. So with that, let me say hi to my friend Ian. First of all, Ian, how are you this evening?
Yeah, I'm very good, Roger. It's beautiful here. You can see the sun shining in through the windows, which is probably the first time you've seen that in Leeds.
With you on this podcast, I think that's right. Now, we've not had Ian on in a while, but he had a rather schizophrenic ending to the football season in that his team, Newcastle, qualified for the Champions League, but his hometown team, Leeds, was relegated. So I was visualizing one of those harlequins with a half smile, half frown, and you walking around town with a face like that for a day so that nobody knew your true feelings. Did that work?
No, I think the joys of Champions League qualification completely overwhelmed any disappointment of Leeds being relegated. Perhaps the greater disappointment was my dad's a Coventry City supporter, and they were in the Championship playoff final and lost on penalties, so that was a bit harder to take.
Yeah, that was crushing. Yeah, I watched that, and that's hard. And then we have Jörn back with us after a couple of weeks. Last time we had Jörn on, in fact, his football team had just denied Dortmund the German championship with a 2-2 draw in a match that Mainz was not expected to do well. How have you been, Jörn?
I'm fine. Coming back to soccer, I think it was Dortmund that didn't score enough to outwin Bayern Munich for the championship. They could have done it. The Mainz team is holding up.
Mainz scored 2 goals in the first 24 minutes, and then Dortmund just kind of fell apart all through the match.
Yeah, they shouldn't have allowed those, I guess. But yeah, I was busy. I'm just returning from UEG scientific committee meeting in Vienna this afternoon. So we're planning UEG week after EASL. There is in the fall the gastroenterologist meeting, and that's going to be full of excitement. So a lot of abstract visualization, and actually there's also some more abstract reviewing rounds going on, and it's really a very busy summer.
I was going to say, you've been about as busy as anybody I know recently, in particular anybody I know who is leading a lot of phase 2B and 3 drug trials or major diagnostics initiatives. You're a part of all that stuff, and then you got a lot of lot else you're doing. You've been a pretty busy guy.
It's an interesting time, I agree, and I'm happy I can contribute on my very own level.
So with that said, why don't we just go groundbreaker? One good thing that's happened in everyone's lives in the past week. Brave one, go first.
I'm going to start because one of the reasons I wasn't on is I came out of a week of vacation. We spent some time in the northeast of Germany, and contrary to what I hear from Ian in the UK, we had a week full of sunshine and clear skies. So time off with the family is always great, very important to me, and so I'm started very relaxed into this week.
Excellent. Ian?
Yeah, well, I think we have had the same beautiful weather, which is absolutely wonderful. And to top it all off, my two older children were away at a scout camp last weekend, which they very much enjoyed, and it allowed me a little bit of time to go on a cycle ride into the Yorkshire Dales, so just over 60 miles or 100 kilometres. But it was beautiful Sunday morning, quiet roads and beautiful sunshine.
That's a nice ride. I guess I'm old, or I'm not in that good a shape. My relaxing rides tend to be about 40 to 50K, but I don't think I can't envision myself doing 101 shot at this point in my life. But that sounds fantastic. We've had, for us, very temperate weather, but it's the same weather you're talking about, which is in this time of the year in Pennsylvania is a decent shot that it's well over 30 degrees Celsius, maybe even 32, 34. We've not seen anything like that. And then I was in Texas last weekend, and it never got over 30 or 31 there. So that was great. And I was in Texas because my goddaughter's daughter, if this was a god-granddaughter, I guess this would be that, graduated high school. And then I came back, and yesterday my granddaughter graduated preschool. So I've seen an 18-year-old graduate and a 5-year-old graduate. Needless to say, they're slightly different graduations, but both were celebrations. And it's just been a wonderful week in terms of families stepping forward. And with that, why don't we get started? This is a pretty simple episode. We've each selected several abstracts from the upcoming meeting that we think are of interest. And you've got the choice of either simply saying, here's what's in the abstract, or maybe not going through it in quite as much detail, but talking about why you find it fascinating. Because Ian is our guest, and we should be gallant about this, I will invite Ian to go first so that his first choice doesn't get stepped on. And we'll go back and forth for 2 or 3 rounds. We'll see what we come up with. And then, as I say, when we're done, Louise has her own contribution she made yesterday. And then we'll go on to the HistoIndex episode, which has no abstracts in it at all.
And you booked us for 3 hours, right? So we get all the shots at the abstracts we love.
No, I didn't do that. We're saving that for 6 o'clock in the morning in Vienna so that we can tape a live session while we're there. And with that, Ian, your floor.
Yeah, well, you've really set us up there, Jörn, haven't you? We can all have an early morning breakfast with Roger in Vienna. So, which I'd be really looking forward to. It's an enviable task, isn't it, getting to go through the abstract book before a meeting? It's one of my favourite things to do because it gets you prepared and interested and ready to go to the meeting. And I don't think there's any doubt, at least in my mind, that the abstract that we're all looking forward to, or at least I'm looking forward to seeing, is the presentation of the Maestro-NASH data. I mean, that has to be the sort of jewel in the crown, and I sort of expect that we'll see the publication of the paper at the same time as we did do in the old days of hepatitis C, with presentations and publications being coincident. And although the top-line data have been read out in press release, it'll be really interesting, I think, to see a bit more of the data, particularly around the potential prevention of progression of fibrosis, stabilization. I know that we've talked about that on the podcast previously, about potential impact for patients, and in light of the OCA experience, a bit more about the adverse effect profile. I know we've seen quite a bit of that through the various Madrigal studies, Maestro-NAFLD, that was presented several times over the course of the last year. But I think it's important to see that safety data accumulating and a bit deeper understanding of the efficacy endpoints. And I guess that there'll be a lot of excitement and interest around that presentation and I hope the associated publication.
And it's actually oral abstract 1 in the general session, so it kicks off the whole meeting and sets the scene.
Yes, that's exactly right, with all the appropriate attendant oohing and eyeing that one can expect to come as a result of that. I think that's exactly right. Ian, I'm not going to say that was cheating, but boy, was that easy. Jörn, why don't you go ahead next?
I guess there's a lot more you could say about that abstract. Well, I remember the Intercept data being presented. That was called the watershed moment, and I think this is the second big phase 3 trial, which is reading out, and the press release data was just even more robust. And after all the discussion about FDA efficacy safety ratio, I'm very much looking forward to also safety data that maybe wasn't detailed in so much depth. So clearly the presentation will go a little deeper there, and that's big excitement. But moving away from the general session opening abstract, I'm digging a little deeper into the abstract book, and you got to love your own stuff, otherwise you would not show it, right? So a fellow of mine is presenting a poster on Thursday today. It's poster 437, and it's called Impaired Health-Related Quality of Life in People with Type 2 Diabetes and Advanced Fibrosis. And it's not rocket science, but it's an application on how we do medicine these days for patients with NASH. So we went out into 2 private practices, seeing patients with type 2 diabetes outside of the university system, and fibroscanned them, and then in addition assessed some questionnaires, health-related quality of life using the CLDQ, among other things, and enrolled 149 patients. The results are somewhat expected, but it quantifies the disease burden we have in those type 2 diabetes practices in Germany. Obesity and advanced fibrosis negatively impacted on health-related quality of life. It just reminds us how important the work is we're doing here, and that we want to offer to them something in the future. So that's a poster. If you're interested, stop by. Again, a very hands-on approach, screening and secondary care, patients with NASH and advanced fibrosis.
So thank you, Arun. With that, I will split the gap between the first paper of general session and a poster on Thursday. First 2 things I want to talk about are oral posters, and this is OS29, titled Analytical and Clinical Validation of AIM-NASH, a digital pathology tool for artificial intelligence-based measurement of non-alcoholic steatohepatitis histology. Before I talk about the paper, the reason I'm so interested by it is that Stephen Harrison, in each of his last 2 appearances on this podcast, has teased the idea that sometime in a reasonable time frame, FDA might accept AIM as a replacement for histology in terms of drug development. And I believe that this, although I've not spoken with him about it, I believe this is a pivotal paper in that discussion, in part because he's the lead author and the presenter, and in part because, for example, the background in AIM starts with a sentence that reads, the gold standard for participant enrollment and endpoint assessment in NASH clinical trials is manual histologic assessment of liver biopsy. This entire study is an effort to look at different kinds of reads and what happens when you use AIM-NASH for AI-assisted reads and independent reads. So the conclusion very simply says AIM-NASH assisted HPs are superior to unaided HPs in assessing NASH resolution. Histologic burden, everything, liver injury, the whole 9 yards. And the idea that these are consistent regardless of what any other form of test shows or any of those different measures. We're now starting to produce data that demonstrates that prospectively a histologic method can be superior in the kinds of things that I believe the FDA has been seeking data on. So I need to understand more about the paper than is in the abstract, but my sense is it is one of the linchpins for the question that goes, can we move beyond histopathology to AI-assisted or even AI-driven reads for admission or endpoint assessment in studies, which, as we've agreed on this podcast for years, will be huge when it happens.
Yeah, Roger, I agree. I think this is an important abstract. It's actually in the Thursday NAFLD Diagnostics abstract session, and I have some more out of that session to be discussed. But I was reading through the abstract. I think I wanted to see some more data. Where's the data actually coming from? It's a big data set, 1,400 over 1,400 patients, and again, all these assessments. So I think the abstract didn't tell me everything that I wanted to learn from it, and I really need to see the presentation.
You're right. I mean, I couldn't tell exactly where the data was coming from, although it suggested, at least if you read the method where it says ground truth NASH CRN scores for clinical validation were determined via consensus by a panel of 3 experts, and then AIM-NASH was evaluated alone. I don't know where the data comes from.
Yeah, it's 3 clinical trials is what they say, 1,500 patients. It's probably phase 2 trials.
I'm looking at this now. You could probably infer, given that there are authors from Gilead, Novo Nordisk, and Bristol Myers Squibb, where the data might have come from.
All right. So that's the stellar data set then.
Yeah. It's not as clear as it needs to be, I agree, but it's certainly, I think, an important planted flag on that particular hill.
Ian, did you see something in that diagnostic sessions you wanted to comment on?
It was a bit about that abstract. I think the ability to include figures with abstracts, I think, is very welcome. The trouble is that people are putting more and more pretty pictures in, and it makes the abstract book harder and harder to read. But one of the big questions is about longitudinal assessment of biomarkers, and that's a study that relates to MRE, and that whole session looks really interesting, and I know that Jörn's going to expand on that in a minute. But looking at sequential measurement of biomarkers to see how progression on a biomarker impacts on future disease progression, I think it's one of the issues that we're still grappling with and understanding, how frequently tests need to be done and what magnitude of change in a biomarker predicts an outcome that is worse or indeed better.
Yeah, and just to say in that session and round it off, there's 2 abstracts that are going to be presented by Mazen Nureddin and Naeem Al-Khoury that are derived from the so-called NAIL-NIT consortium. Now, that's an interesting consortium I'm a part of because it combines data from completed clinical trials, the baseline data set at least. So for the NAIL-NIT, we combined 6 different data sets, and I agree we didn't say which studies and trials that were in our abstract either, so can't complain about the previous one where they had 3. The 2 abstracts here is is SO-227, and it's the performance of vibration-controlled transient elastography in identifying at-risk NASH from those 6 clinical trials. And again, being embargoed, we shouldn't be discussing it online yet.
Jörn, I'm only laughing because from the title, it was probably my second choice paper, and then I saw it was embargoed.
Again, being a co-author, I've seen some of that, but it's not something to be discussed. Anyways, I think it feeds into what Ian said, that we're seeing more robust data sets of well-controlled, prospectively collected, closely monitored data on the value of NITs in staging the disease, cross-sectional, as it's done for the O27. The second abstract here is the one being presented by Naeem Al-Khoury, and it assesses the accuracy of the AGA referral pathway. So that will be very interesting too in the same data set. And again, as you said, Ian, I think the outcome prediction with the MRE is something that came from Japan, also raised my interest. And I think that's how we use NITs in the future, right? That's how we want to use it. We're going to use an NIT to stage the disease. We're going to use an NIT to come up with the indication for therapy. And then we use an NIT for treatment response. And it could be very different NITs. It's most likely not the same one. And it could even differ between MOAs, and at least when you're discussing treatment response. So this EASL Congress leads us one step closer to how we're going to be managing patients with fibrotic fatty liver disease. We should be saying steatotic liver disease also, Ian, I believe, at one point. And that's, of course, also something that's being discussed at that meeting big time.
Of course, then we're going to have to flirt with whether we have to rebrand this podcast. But that's a whole different headache that we will deal with after the event is over.
Well, from 5 to 10 on Saturday morning, I think that's the launch time. So you have to be ready with your new logo and tagline.
No, we're just going to stay with where we are for a while. We'll get through this meeting first, then we'll worry about it. But go ahead.
Thinking about how we use NITs in the future, there's an interesting abstract about prediction of outcomes post hepatitis C cure by transient elastography 1 year after treatment, which, although it's not directly relevant to NASH, it does give you a sort of window into that future prognostication with treatments. The treatments are very different, but it shows that those values retain prognosticability, albeit at less risk per given liver stiffness into the future. And there are some things that we shouldn't take from hepatitis C, I think, and that is the ideas about short duration curative treatments. But there are many other things that we can learn about how to select patients for treatment using prognostic tools, and indeed how those patients can be managed afterwards. I don't recall the number of that presentation, but it's the first authors, Georg Semmler from the Vienna group, and I know that they are very much looking forward to hosting the meeting in their home city.
I think we have to move on from the NITs and actually talk about the next abstract session. And the next one that obviously sparks the eyes, if you move outside of the general and late breaking, is the NAFLD therapy session, which is on Friday morning, 8:30 a.m., so early rise. And there is 5 presentations, 4 discuss pharmacological interventions, and actually 1 presentation of a young investigator is related to time-restricted feeding. This is a young investigator from India. The concept is very interesting, and I want to touch it briefly because we're doing a similar study. A fellow of mine who's working in Italy, in Turin, actually has done the same thing. Here in that abstract, it's oral abstract 064. They have 68 patients in time-restricted feeding. And I think the first gap in this is to keep the patients on those strict dietary requirements with 8 hours of feeding and 16 hours fasting. And for most of my patients that I approach, that's not feasible because of work shift or something. But if they're successful to stay that, they have a number of non-invasive tests they're assessing. This is actually mostly done by FibroScan here. They're calculating fast. Obviously, the time-restricted feeding was better than calorie restriction on liver stiffness and CAP improvements. So I find that very interesting. I find it interesting because it's plausible and it's easy to explain to patients. All other dietary interventions are complex and more difficult to detail. If you tell them you eat then and you don't eat then, that's pretty straightforward. So I'm a fan and I actually advocate for that for some of my patients. It's not an intervention that is aimed at weight loss, but it restores insulin sensitivity. So for that reason, very interesting.
Yeah, that's fascinating. I have friends, social friends, who've attempted that regimen and argue that even if you can't be completely successful every day, one is a senior vice president of a Fortune 100 company and does not have that kind of control of her schedule. But she's lost significant amounts of weight. I'm going to guess maybe 15% of her body weight in a 6 or 8 month period, and has seen her HbA1c is driven below 6. And she's had a couple of other things that suggest that she's really had tremendous success with it. Now, that's an anecdote. But the only reason I mentioned that actually in context is that that's with imperfect execution. I'm not sure you have to hit the letter of the law on this one to get most of the benefit of it, would be my only question. I don't know how one proves that in a study, because that assumes that, well, I don't know how one proves that in a study, but I'm not sure you need perfect execution for this to be a successful strategy.
Ian, do you have anything in that treatment session that raised your interest?
Well, I think there's quite a few interesting abstracts in that session. Some of them are sort of relatively familiar. The first abstract, OS-060, which is the phase 2A randomized active comparator open label study to evaluate the efficacy and safety of Merck's new drug, which has a name that I haven't heard pronounced in public and almost.
You have to say it. It's ephenopektitide.
Well, it sounds like you can't say it, Jörn. This is a
Ian, no guts, no glory. Well, no, what, ephenopektitide? Pegtitide?
Yeah,
sorry.
Pegtitide. There you go. That'll do. I mean, this is the sort of direction we're headed. This is active comparators. So this is a combination GLP-1 glucagon receptor co-agonist, a bit like one of the other drugs later on in the session. And it's in comparison with semaglutide. And one of the other abstracts I'd quite like to talk about is one that Jörn's been involved with, looking at real-world, or not quite real-world data, but other non-NASH trial data for semaglutide, because I think it's inevitable, likely, that many of our patients are going to be treated or will have received treatment with semaglutide or another GLP-1 receptor agonist, and trying to understand what the additional benefits of treatment are going to be is going to be important, particularly as drug development continues and perhaps the regulatory pathway might change with licensed drugs. So I think beginning to see data like this is really important, and this is an early phase study, but they report really quite striking rates of reductions in liver fats by MRP-DFF, and it's greater reductions with the co-agonist rather than with simply just the semaglutide. And we'll begin to see what this will look like. I think as a sort of headline figure, the proportion of people with a relative liver fat reduction of more than 70% is itself nearly 70% with the co-agonist, compared with 12% with semaglutide alone. So it's these are quite striking results, and I think it'll be really interesting to see how that reads through into 2B and then into later phase studies perhaps.
I agree, and I think we're following the obesity field here a little bit, and we're seeing how much more potent those dual agonists are. And the second one, the pemvidutide, is also a dual agonist. Now, if we start to discuss them, it's important that we remember that they have different agonism or strength of binding to those receptors, albeit they're activating both the same receptors. The ratio of GLP-1 over glucagon is important for the effect size and also for side effects potentially. So, albeit in the same class, I think the profile could be very different, and that'll be very interesting to learn. So also always have to look at the safety aspects of these. And I mean, the other two mechanisms that are discussed in that session is a FAS inhibitor, and we've seen some of that compounds earlier. So Rowe and Jörn abstract 061 is going to present data on denifanstat. I'm not sure that was 100% right, but it's interesting. Only 26.
Close enough.
Close enough, right? 26-week interim analysis of the FASCINATE-2 trial. The FAS inhibitors are a class that keeps promise. And then a very new MOA with the RNA interference. I think that's fascinating because it's really down to individualized therapy here, and we're seeing in other indications the applicability of this. And here, targeting the hydroxysteroid 17 beta dehydroxygenase 13 is really novel, I think.
That one I have a couple of points about. First of all, one of the last things I did before I left marketing research was the global forecasting and benefit strategy for inclisiran, now known as Leqvio, which is like this molecule, a siRNA dedicated for a specific target, in that case PCSK9, and developed by Alnylam. And Alnylam marketed with the Medicines Company, which was eventually sold to Novartis. This is Alnylam and Regeneron, but fundamentally very, very much the same concept. Okay. Second, Jörn, I don't think you were a co-host yet, but at one of the meetings in 2021, Mike Charlton suggested that he thought the most interesting abstract in the entire meeting was a late-breaker poster for an Arrowwood siRNA that subsequently had its development discontinued. A month later, Novo bought a company that had its own siRNA. So we're now starting to see this stuff bear fruit. And then we had an episode with Scott Friedman where he actually talked about the idea that they now can put miRNA together with CAR T and go directly after fibrosis that way in an individualized way. This is, I think, one more early shot in what's going to be a fascinating set of advances. You can really hit a target dead on. And CAR T became an issue in oncology in part because you're using externally generated material. But here, you don't have to take anything out of the individual. You can just target the gene and go. I expect we'll see a lot more of this in years to come. And I agree with you. I think it's a fascinating area.
Yeah, the limitation of it is a little bit, you always have to, in these earlier studies, they show target engagement in the liver, so you still need liver biopsies. It's not, to also show you knocking it down. And I'm not sure how long we want to continue this, but it's interesting to see how strong the decrease even in liver tissue will be.
I think actually the benefit of the therapy will determine whether we continue doing biopsies. Once we've got drugs in market, that's going to become a lot less attractive for everybody. And once NIT is accepted for drug approval, even more so. But I would guess that there will be a way around it for a therapy like this because this stuff has some really interesting potential to it. On the other hand, I'm not a hepatologist, so I'm like the last person who should render an opinion on that.
Roger, I think it'd be interesting to see if there's any sort of biological signals of activity that can be identified in something that isn't liver tissue. They'll be looking hard for that. And I imagine, and even if it's just ALT or AST, something to indicate activity beyond, target engagement on the biopsy. But I guess we'll see. I agree. I think it's a really interesting presentation. Very, very early phase in that session, which is a contrast to everything else. And I share the interest, perhaps a bit less the excitement, because I think, the way the field's moving, I'm not sure how it would fit in the sort of therapeutic environment in the future. But I think the biology is really interesting, and understanding whether you can have a therapeutic benefit with that sort of strategy will be really interesting.
Well, so there, Ian, I think we can actually steal a page from cholesterol, which is that when we got done doing as much good as we could do with small molecule, PCSK9 was identified, and 2 agents targeting PCSK9 more directly came to market with Q2-week dosing and very, very high development costs. And Leqvio is Q6-month dosing and is not quite as efficacious against cholesterol. I think something in the range of 50% versus 56 or 57% drop after high-dose statin. But if you can do that in 2 doses a year in patients where you've already worked your way through all the small molecule therapies, there should continue to be a place for that, I would guess.
Yeah, I totally get that. I think it's about biomarkers for efficacy and response, and perhaps even mechanism-specific biomarkers. And I think understanding how we will gauge treatment response in individuals, and whether we will have response-guided therapies, or whether these will be therapies that we'll use in the longer term. They're still open questions in my mind, and it speaks to the amount of work that's going to be required if and when a drug is licensed, because there will be so many questions still open about how we actually use these drugs in practice that keep us and the podcasting business for many years to come.
Yeah, that's exactly right. I made a comment on one earlier episode that in 3 years I might not be on this episode. Jörn reacted with alarm. So I guess I have to be here for more than 3 years, Jörn. Yeah, I think this research will be ongoing well past when my 5-year-old actually graduates high school, 5-year-old granddaughter, and that'll be many years hence. Next topic to move on to, gents?
Yeah, I'm just following the abstract sessions. I'm not sure you wanted to highlight something specifically next, Ian, but there is a NAFLD clinical aspects on Friday afternoon, 5 till 6:30. It's chaired by Nicholas Goossens and Hannes Hagström. Very clinical presentations, five presentations. Real-life data, use of anti-diabetic and lipid-lowering medications in the U.S., risk stratification, MEFIB, MRI, PDFF. There's another digital pathology, stain-free imaging, the prediction, and here you go with the actually prediction of outcomes. It's presented by Timothy Kendall from Edinburgh. Then there's a SEMA paper in there and real-world data from the target NASH. So I think this whole session is very close to what we're thinking today when we see patients. Can I treat him with semaglutide? What's his risk? Will he decompensate? How can I risk stratify? This is, which drug that I have available should I use to manage my patients? This is for somebody who sees NASH patients. I think this is all covered here.
I agree with that. I thought it was a really fascinating session.
Yeah, I'm looking forward to seeing the Edinburgh data. I've not seen it in development, but I've been talking to Tim Kendall and John Fallowfield about it intermittently over time as a sort of interested observer. And they've accumulated a lot of biopsy samples from across Scotland and have put them into a common data warehouse. And that's allowed them to do some research and to think about prediction of future events and to think about biology too. But it also is in a sort of common data commons so that they will, so that data is stuff that's available to other people to use. And I think seeing it out in the wild for the first time will be really good.
Yeah, if we stay with that abstract and it's oral presentation 087, the nice thing is that I think it tries or it aims to advance a segmentation of fibrosis beyond those 5-tier staging systems we're using, right? The 0 to 4. And there's actually, they mention 184 fibrosis parameters from a single unstained slide. And that's, of course, a lot more than a pathologist could do. Potentially, they're all included in our 5-tier system. But I think, not that we want to use that in clinics in the future, but it will help us to better understand progression, regression, and what fibrosis is, how it's behaving, really.
And I think think that's right. One of the things that's missing in the sort of digital path AI fibrosis assessment, at least, is an understanding of what changes in these various parameters that are being described mean, and we have a sort of reasonable understanding of what a change in fibrosis stage means or might mean to a patient. But I think seeing cross-sectional outcome data, at least for some of these parameters, will help us begin to understand what a change in. We definitely need that if we're going to move towards digital path or AI type guided or made endpoints in trials.
I've got two other things I want to make sure we cover, one of which I'm going to put Jörn on the spot a little bit, the other of which I'm just going to cover. So Jörn, you're chairing a session Friday afternoon, right? I was hoping you could talk a little bit about it.
They were more generous with me. I can have my glass of wine after my session, and Ian has to behave on Friday evening and get up early on Saturday.
By giving Ian a 6 o'clock session, we're allowing him to control his wine and blame a third party. It's a wonderful thing.
So this is not abstract-based. This is an interactive session. I guess they were called earlier Meet the Experts. Now it's more of an interactive discussion session. I'm running this with Elisabetta Buccianesi from Turin. They've done some great studies together with the Newcastle group and other colleagues on lean NAFLD, and we'll be joined by, among others, Shira Salvasaggi as a discussant to, revisit the concept of lean NAFLD. To me, it's always a very broad bucket, and we are combining a lot of patients there. The lean NAFLD, I think, is more diverse than the other typically metabolically burdened patients we're seeing, and we'll be discussing some of those pathophysiological aspects, the role of genetics, and then we have Shira there, and she'll be, of course, detailing management of lean NAFLD, because that's not easy. I mean, the drugs we've been discussing here, the weight loss and insulin sensitizers, they could help a little bit, but of course, that's not the main driver of their disease there, necessarily, overweight and obesity. So the German guidelines, we actually just speak about physical exercise for these patients, and I think we can do a lot more. And if you're interested, come join us. We're open for the discussion, and we have great faculty to lead us through the session.
That sounds fantastic. There's also a session Friday at 3 that I don't think I'm educated enough to handle, but I'm kind of hoping that someone will handle and then come on this podcast and talk about, which is the role of miRNA and LNC-RNA in liver disease, where they actually have 3 papers with Q&A, one talking about small and long non-coding RNAs in liver cancer, one talking about long non-coding RNAs and RNA-binding protein complexes in HCC, and one talking about non-coding RNAs in non-malignant liver diseases. If some of the future is going to be in that stuff, this should be fascinating, even if we're early on. It's being co-chaired by Sven Diedrichs from Freiburg, Tushar Patel, and I'm not sure exactly what will come out of it, but I'm pretty fascinated. And then with that, let me ask you guys, before we go to final, anything else you want to comment on? Ian, other papers, other things you're hoping to get out of the meeting, besides a good, decent wine on Friday night and a hellacious Saturday morning? What is the session? So tell us, what is the session you're chairing at 8 o'clock on Saturday morning?
Oh, so on Saturday morning, I'm chairing a Meet the Expert session. Not chairing I'm running a Meet the Expert session with Peter Jepsen from Aarhus in Denmark, and we're talking about early diagnosis of liver disease, and it promises, at least according to the questions that we've written, to be a really interesting discussion about, target population for early diagnosis testing or screening, what tests you would use, what interventions would follow those tests, and then there's a really provocative final question that people can think about before they come to the session, and that is, what happens if you set up this testing or screening as you think it should be, you implement it, and then it doesn't work? Why would that be? The pre-mortem of the project. And it'll be a good session. There's a lot to talk about for an hour, and we are sure to have a wide-ranging discussion about the value of screening for liver disease in the general population or in selected populations, depending on what people say.
Fantastic. And then, Jörn, is there anything else you're chairing or sharing at the meeting that you'd like to go over with us before we go to final question?
No, again, there's a number of presentations I'm co-authoring. I'm very excited about the big diagnostic and LNIT data, and I think they have 5 or 6 presentations. One is going to be given by me as a poster. Also some novel stuff derived from actually the SEMA weight loss step 1, step 2 studies, looking at some novel biomarkers, somologic proteomics there. So I think a lot of things to look at and do, and really the start of a big step in improving our understanding of the disease, pathophysiology, treatment, diagnostics. Very exciting, EASL.
I agree, and I share a thought that Ian mentioned possibly before we went on air, possibly after we started recording, about packing all this into fewer days than before and having so much more to do. This meeting could take 4 days and give you time to think about everything between events, but I understand Alexander Krat came on and explained why they went to 3 days. It makes perfect sense. It's just going to give us a higher level of brain exhaustion by the time we're done, I think. With that, any closing comments about the meeting?
One abstract I stumbled across, again, because the general sessions are the ones where you have the high highest ratings. In the general session 2, there's actually an abstract on bariatric endoscopy, weight loss, and the effect on steatohepatitis, endoscopic sleeve gastroplasty. It's general session 009, an effective treatment in steatohepatitis, prospectively multi-center randomized trial. The abstract is under embargo, so not much to be discussing, but the urgency to tackle this disease, and I think we're hopefully going to move away from surgical or let's say suturing techniques, might still be of need for a subset of patients, but it shows how big the need is to develop effective drugs. And the drugs hopefully will come into the arena of weight loss that these procedures can achieve in patients.
Agreed. Because we've got so much to pack into this episode, this and then Louise and then Histo, I'm not going to go into it in detail, but I might put out a note, or I might talk on our episode next Monday, which is mostly going to be about ADA, about a couple of posters that I thought were particularly interesting. As I say, we've got plenty of ground we've covered so far. So I look forward to seeing you gents next week. Ian, you're going to get an invitation for Saturday morning at 6 o'clock now that you've semi-volunteered to do it with us, and that'll be exciting.
Yeah, Roger, your persistence has led this Surfing the NASH podcast to maintain over a number of years by now, so that's the way to carry it on.
Of course, burnout is always a possibility for all of us, but I think so far we're rolling along just fine. So I look forward to seeing you folks. Next Monday we'll be talking about an ADA preview, since that happens to be the same weekend in San Diego, and we'll have a lot of, if you will, hepatoendocrinologic work in it that we'll want to discuss. We'll have two sessions we'll record while we're there, I'm hoping. And then we'll be back after that on the 26th to talk about what did we just learn, with a slightly different cast of characters in this one, although Jörn will be part of it. Louise will be there. I believe Quentin Anstey will be there. I believe Stephen Harrison will be there. So that should be a lot of fun, too. With that, let me thank you, gents, for your time. I will be next with Louise. I don't think we'll do a business report at all today because we have so much else in the podcast. But safe flights or travels, however you get there. And I look forward to seeing you, gents, next week in Vienna. Thanks.
Thank you. Bye-bye.
Bye.
Bye-bye now.
In our next session, Louise Campbell and Roger Green discuss some of Louise's highlighted abstracts for the EASL Congress 2023.
So, as promised, we're following up our episode with Jörn and Ian with Louise, who's with us this afternoon. Was not able to make the full taping session. So we're doing this as an interview, and in fact, she will be virtual at the meeting as well. Hey, Louise, how are you doing today?
I'm very well, thank you very much. Nice to be with you on such a nice day in Somerset.
It's a nice day in Somerset. It is a rainy day here, but that's not a bad thing. We've not had a rainy day in a while. It's very rare that one gets excited about hailstones, but some of my neighbors might be. So you're missing the weekend live because you've got big family goings on, eh?
Yes, everybody's flown in from around the world for my mother-in-law's 90th birthday, which just happens to be obviously the week of the reshed. Now that EASL has moved into more summer months rather than the previous Easter timing. So most other years I'd be absolutely fine to be enjoying Vienna and meeting up with people again, but sadly,
and I will actually be there despite the fact that I have a family conflict also, but I can't cover the meeting if I'm not at the meeting. I'm going to have Mike Patel helping me, and we've got some KOLs lined up, but I kind of have to be there for that. So we're going to take the next bit of time and talk about a couple, 3 abstracts that you find particularly interesting and that you will be able to hopefully talk about when we do episodes. Where shall we go first?
Well, I think I'm going for a poster presentation to start with, and it's on the Thursday, and it's 414, and it's by Ying Shang, who's presenting it, and the group, and it's Causes of Death by Fibrosis in 959 biopsy-proven NAFLD patients. Sterling title.
By the way, that Hannes is one of the people who will be joining us over the weekend, and this is obviously his group.
Well, he's got a number of abstracts in from my flick through the book.
Yeah, they publish a bunch of stuff, that unit.
To be fair, I thought this was an interesting article for a couple of reasons, and ultimately, the backgrounds and aims of it were the causes of death in patients with NAFLD. It may differ on the fibrosis stage at diagnosis. So they wanted to aim to assess the distribution of different causes of death through the group, but they were all biopsy proven, which is obviously the gold standard that we need at the current moment in time. So they conducted a retrospective analysis of 959 biopsy-proven patients from 3 university hospitals in Sweden between 1974 and 2020, and the causes of death were ICD-10 coded from the Swedish Causes of Death Register until the end of 2021. So liver-related deaths were defined as hepatocellular carcinoma, cirrhosis, decompensating cirrhosis, and liver failure. So the results of that, among the 959 patients, the mean age was 49.5 years, 61.7% were male, but 335, which was 34.9% of the cohort, died during the median follow-up of 17.9 years. So it was a long study. Mean age of death was 74.6 years, and 52.7% were male. And of those, 75 patients had zero fibrosis, 120, which was 32.4, had F1, 32.7% F2, and 45% of patients were F3, with another 47% of F4, and the died of...
Is that Louise, is that percent of population, or is that the percentage of patients at that fibrosis level who died?
I think it's that fibrosis level is the way I read it when it says the mean age of death.
I guess my thought is I'm looking at these numbers, and I got 5 numbers that add up to a couple of 100%.
Well, I was looking at that at the same time, so I think it's 26, and patients, for example, in F4.
And that the cohort would wind up being 53 or 54 patients that they were observing in F4. Okay, got it. Okay, good.
And I think that's the way it's meant to, because yes, when you add up those numbers, it's certainly not 100%. So when I was looking through this piece, what struck me at the end was something we talk about here a lot, is that the conclusion in the ultimate study was that patients in NAFLD and cirrhosis at diagnosis, liver disease is the most common causes of death, which is what we've discussed in the past, that where you look at liver disease is the cause. But in patients with no or non-cirrhotic fibrosis, cardiovascular and extrahepatic cancers were the biggest cause of death. So different management strategies need to be employed, was their conclusion for that. The reason I was interested in that, we discuss it here a lot, that do we look for purely liver-related outcomes and liver disease, or do we start to stratify NAFLD by the metabolic cardiovascular risks rather than just the NAFLD and liver that we look for? The evidence that this was coming out with sort of strengthened my views. And so
I get that, and I had the same reaction. It's interesting that they put HCC in with other liver-related causes of death. So at first, when I first looked at this, my reaction was, well, gee, where's liver cancer in here? And then I realized, but what about, I'm eyeballing here about, 55% of the F4? Now, you're not dealing with large populations, but F4 is significantly more likely to have had liver-related cause of death than anybody else. Once you get past that, I'm not sure those other differences are significant.
Yeah, I agree with you, but I think it's this, if we just screen for liver damage and liver disease, that yes, we're going to find liver-related mortality in that population more so. But actually it broadens that whole question of who are we looking for when we look for poor liver health. Because in this study, it is those that didn't have cirrhotic NASH, people we would discharge back to primary care, ultimately, if we were just looking for liver disease, actually had higher rates of death in other areas. That, again, we discuss an awful lot about this cardiometabolic link to type 2 diabetes, non-hepatic cancers, and cardiovascular disease. So I think it's liver health for me. It's liver health, and this is a screen and some evidence that liver health counts as just as much as liver disease.
No, probably more, actually. If you take a look, there are slight increases in the liver-related deaths, but you're under 20% for F3 and downward from there. Interestingly enough, F0, F1, you have other as a significant cause of death as well. But that's not surprising. All right, so that makes good sense. I'd love to know what else they know about these patients that they'll be able to tell us in the presentation, but I like this. This is interesting.
I'd like to, obviously, have been able to see non-invasive technologies used to assess FID4 and things like that, to see how they screen. But these were all biopsy-proven patients. They've got a great data set and cohort in Sweden, and Hannes does some great work.
Hannes does fantastic work. What other poster or paper would you like to cover? Just before we do that real quickly, for those who are looking, that could be found on page, I guess it's 631 of the abstract book. So if you want to scan for that, THU14 found on page 631. So which other are you looking for, and which page can we find it on?
Yeah, my next presentation that I chose was THU, which is Thursday, and it's 410, and it can be located on page 629 of the abstract book, or S603 on the bottom corner. And it was a systematic screening of NAFLD-related advanced fibrosis high-risk population in diabetology using transient elastography, a prospective study. And this is being presented by Cyril Corsi. She's obviously been on the podcast, and her strength and the team's strength is the diabetes. So the background aims of this was the systematic screening of NAFLD-related advanced fibrosis currently recommended in high-risk populations such as patients with type 2 diabetes and or obesity, although there are limited prospective data from patients enrolled and systematically assessed using transient elastography and diabetology. So they aim to examine the utility of non-invasive tests and the prevalence of advanced fibrosis fibrosis in this prospective cohort in the diabetes. So it was a multi-centre prospective study, NAFLD care, including patients with type 2 diabetes and or obesity, average age between 40 and 80 years, BMI of less than 40, enrolled in a systematic screening, advanced fibrosis in 4 diabetology centres in France between October 2020 and November 2022. So all patients had a standardised research visit, fasting labs, including fibro test, liver assessment by FibroScan, and all other causes of liver disease were systematically excluded. The presence of advanced fibrosis was determined after the assessment in hepatology with either histological fibrosis stage of greater than 3 or equal to 3, or overt imaging diagnosis of cirrhosis, or concordant transient elastography of greater than 8 kilopascals, and a fibro test of greater than F3 according to the EASL guidelines. So of the 487 patients screened, 461 met the eligibility. Mean age was 59.5 years. BMI was 32.7. 46% were females. 75.5% had obesity, and 87.4% had type 2 diabetes. So 72% had dyslipidemia, 67% hypertension, and among them 55.5% had a FIB4 of less than 1.3. And the proportion of patients with transient elastography of equal to or greater than 8 and equal to or greater than 12 was 18 and 4.8 respectively in those groups. So they ruled in and ruled out advanced fibrosis in 71.4% of participants based on the transient elastography of less than 8 and a fibro test of less than F3 in these diabetic patients. So I think as they went through, they looked at 82 participants had determined status of advanced fibrosis, 27% confirmed diagnosis with advanced fibrosis, and 54 undetermined status of advanced fibrosis due to discordant non-invasive technologies, which can happen, and no liver biopsy results. So advanced fibrosis for 6.6% of the participants, and a higher BMI was significantly associated with an increased odds ratio of 1,114 with a 95% confidence interval, 101 to 1.24. But adjusted for age and sex, FIB4 of less than 1.3 had a high negative predictive value, which we would expect, of 98%, and among participants with advanced fibrosis, 15% had false negatives. The conclusion of the study, the estimated prevalence of advanced fibrosis in this multi-centre study in high-risk populations was 6.6%, as we've just said there. But what they were able to do was the high negative predictive value of advanced fibrosis, but this significantly misclassified. A FIB4 of less than 1.3 had a very high negative predictive value for ruling out AF, but significantly misclassified patients younger than 60 years of age. So that age range coming into changing the outcomes of these patients and missing them. So further analysis in a larger population is key, and I think this study, it was about trying to get the right patients. We've all looked at FIB4 and looked at different age groups and where it has weaknesses, and particularly in younger ages, but this inability to look at the false negatives, I suppose, in that group. We are doing better than we were before by using it. So the conclusion was all about FIB4, not necessarily about transient elastography.
The thing I'm struck by in the abstract is not just the cut edge of 60, but the mean age for the participants with the negative predictive values versus those with correct positive prediction was 51 versus 66.6, which is really quite a difference. So they set it at 60, but it feels like the younger you get, the worse it gets. Yeah. I'm trying to figure out why that is. I mean, well, I guess, we know Sarah, so we can ask her. But my snap reaction is that FIB4, as Stephen and Mazen have both talked about in different episodes, FIB4 performs worse when you're dealing with highly enriched populations, because it does not do a very good job on positives, right? So you're seeing that again here, high false negative rate. But this is not particularly, doesn't feel like an enriched population per se.
It's our low-hanging fruit, isn't it? So it had a great ability to rule out patients, and I think that's the really good thing for me. We don't want to get lots of people in the wrong category. So its ability to be a predictor of who wasn't going to have it was great, irrespective of the weakness.
I guess that's right. And missing 15% of those people, as they point out, 15% at this point comes out to be an N of 4. So that's interesting, because I think that ultimately the challenge around FIB4 is how well does it do as a first-line screener, where the challenge is entirely around false negatives. We know it's very good at negative prediction. The question is how many false negatives are we going to get, because those are people that we wish we were treating. And is there any way to figure out who they are? are. This obviously doesn't get to the second question. But the question, I guess, is 15% false negative an acceptable rate?
I suppose the question for me is, if we were to look at the AGA guidelines and that, we'd be looking at an indeterminate group of above 1.3 and below 2.67 who should have the FibroScans. And I know they did FibroScans, so I'd like to have seen that more granular breakdown of the FIB4 and how it functioned in the diabetology population in those categories that we use. But that's not evident here.
That's a great point. And then the only comment about that is that when you got an N of 4 that are false negatives at 1.6, if you cut it at 1.3 and then at 2.7, 2.67, you're going to have, 2 on one side, 2 on the other side, 1 on one side, 3 on the other side. Sample's probably a little small to support that level of analysis. It's absolutely the right question. Red, yellow, green works a lot better than red, green on this kind of thing.
And that was an outcome, wasn't it? It was a bigger study should be done with a bigger population. And hopefully that's what the team are doing. And probably people around the world are doing that in different countries for their diabetes populations. Low-hanging fruit, as Stephen calls them.
I'm sure that's right. Anything else you'd like to talk about today? We could probably take another 10 or 15 minutes if you wanted to. If you have any other papers that you want to talk about or general impressions from having looked at the essay.
I do have another paper.
Let's go. Which one is that?
Particularly on the back of all of the things that happened last week with the publication of the NICE guidelines for FibroScan, but also International NASH Day. But this abstract is more in general for liver disease. So my next abstract was page 903, which is FRI, so Friday, and it's 118. And it's patients with persistently abnormal liver biochemistry are under-investigated and can be rapidly identified using novel case-finding database. Now, this is being...
This is PHI, right?
Yes, this is being presented by Al-Mathana Mohammed, but it is from the team that we've had on here, Tim Johnson and Predictive Health. So the background aims of this, which is why it's important to us, because NICE have obviously now delivered on putting FibroScan into primary care and outside specialist and secondary care locations, more importantly. These are the exact questions we want to be able to find. We want to be able to locate people and then scan the highest risks so we can risk stratify those patients. So the background aims of this, with chronic liver disease, continues to increase in prevalence, but it remains underdiagnosed, which is what we're trying to address in the UK. So many patients are missing opportunities for treatment. We're missing opportunities to get them treated. Guidelines state that patients with persistently abnormal liver chemistry should be given a non-invasive liver screen to identify potentially treatable causes. So this brings us in with John Dillon's work with ILFTs in Scotland, for example. You would be able to do that. So unfortunately, in practice, these guidelines are often forgotten.
So the data that they're using, right? They talk about ALT levels and ALP levels.
So they're not followed and they're developed. So they developed the novel case finding database in Somerset, where I'm sitting now. So it's a little bit personal. So they've got a data on 560,000 individuals. That's probably near enough the entire population of Somerset, to be fair. So they use the system to identify patients with persistently abnormal liver chemistry and quantify the completeness of this investigation. So these are all guideline recommended. All of these patients that they detected really should be followed up if we're talking about implementing. And I suppose it brings me to any future pathways we develop. How do we ensure compliance? Because we don't get compliance currently in a lot of these cases. So using the data up until the end of December in 2020, so 31st of December, I'm sure they weren't working over Christmas, but the case-finding database was configured to identify patients between the ages of 30 and 75 with persistently abnormal liver chemistry. So last ALT was greater than 40 international units per litre and abnormal for at least the preceding 90 days. Within the cohort, they further risk stratified to identify those with concerning results: ALT over 80, ALP greater than 90, ALP greater than 130. So they split it within the groups: ALT greater than 80 and ALP greater than 130 for both. The number of patients in each group with a complete basic non-invasive liver test, which was determined by viral hepatitis B and C screening, autoimmune screen, ferritin and AST, was then looked at. The screen was considered complete if those tests were found within a 6-month period of the abnormal liver tests. So based on the ferritin data as a commonest test. A sample of cases were reviewed manually to confirm the accuracy of the results, so I think that was quite thorough. They also assessed the age cohorts where they're likely to be persistently abnormal abnormal liver chemistry. And if there were any differences in age, likelihood, having improved the basic non-invasive liver tests. So they got 8,224 men, 2,537 women were identified. So we're only a small county, Somerset. Only 11% of males and 16% of females had complete basic non-invasive liver screening. So that is really concerning, because there's another 89% of men walking around in that population who are at risk, and potentially another 84% of the women in that group.
That's 5 out of 6 women and 8 out of 9 men.
Yep. And we wonder why we're having a problem with the disease.
And we're really talking basic here. We're talking about ALT, ALP. That's pretty basic.
And we're not talking about alpha-1 antitrypsin. We're not talking about hemochromatosis screening. We're talking about just a fundamental non-invasive liver screen, set of bloods. So there were no significant differences identified in the likelihood of having a non-invasive liver screen when they looked at the different age groups, which were 50 to 59 cohort in men and the women. And in the higher risk group, abnormal tests at least 90 days, the last ALT of greater than 80, last ALP of greater than 130. There were a total of 547 patients identified, of whom 442 had never had a non-invasive screen. So 81% of the high risk group had never had a screen. So there was a slight trend towards improved investigation in higher risk groups that was not statistically different, and they show that in their figures. So the conclusion of that, as we're probably making out here, was that the data confirmed that patients with persistently abnormal liver chemistry are frequently not investigated. I think frequently paints a nice picture on it rather than how severe it could have been.
You could go for virtually never, right? You could convert frequently not into virtually never.
Yes, so those who just get one abnormal test have got practically no chance of ever being followed up, if we're to believe this. So our novel case-finding database can rapidly identify in seconds nearly 9,000 individuals who may benefit from further investigation. Furthermore, the system could be used to easily stratify, and I think it's the ease of being able to do this, because when Tim and Lyd came on to discuss it, they backread the serial results, and what they're doing is picking that trend up of these patients becoming more and more high risk over the period of time. But this abstract is really, it's a poster presentation, but it really sums up why we have a problem with late diagnosis. Because we rely on blood tests. We all want a blood test to be able to predict whether it's FIB4, whether it's any of the ELF, whether it's any of these. But if we cannot pick up the basics to even trigger more investigation, then using a blood test isn't necessarily going to be the right thing. We've got, as you say, 4 in 5 women.
5 and 6 women, 8 and 9 men.
Yes, falling through the net. And we're never going to get on top of late diagnosis while these people are presenting at that level. And I think that's the concern. And this highlights this for me. This is just an English population in a county that would be considered predominantly white. It's not an inner city region. It has its own problems with drug and alcohol and hard to reach populations, but it wouldn't be your complete mixed ethnicity demographic. Now, there is no data here for that demographic and the mixed sort of what were the cohorts, what was the ethnicity mix in here. But knowing Somerset, it's probably not going to be that diverse.
And this is where Tim did a lot of his work, right?
Yes, this is where he currently practices. And so this is why the strength of this information, they've done this very well.
I'm hearkening back, as you teed this up in the context of last week, I'm hearkening back to Will's reaction to me saying, I wish we had ASTs here. And he said, well, you're just not going to get those. But this is enough to start scraping. And you would hope that with more FibroScan centers in place, and with a nice reimbursement, you'd be able to get people from here to VCTE pretty quickly. Yeah,
we're going to develop that reimbursement process. There will be a time lag, but I think if you can't get people to FibroScan, you do also need the basic liver screen, because if you're going to say that FibroScan is going to be relevant, picking up on your conversation and what you were talking about last week, you can't put fast in if you don't have AST.
Right.
If we are missing this amount of the population, we cannot recruit to trials. So pharmacological companies and trial centres do have an obligation to try and look at how can we improve what we're doing to find the right people to trials. Because if we could get this cohort that are being missed, screened and into trials, we could nail liver disease quite quickly. Because we're missing most of them. We need more people into trials.
Let me think about this for a second. So if you could get to 50%, you would have trebled your female trial population and somewhere between quadrupled and quintupled your male trial population.
Yeah, by just helping primary care
right?
get the right tests into the right people.
And as we note, that's in a country that may look harder at liver disease than many others.
Yeah.
Well, okay, Louise, I'm thoroughly sobered. So thank you for that. I think all those points are important and valid. And I can see it has turned to night behind you in the half hour we've been talking. So let me let you go have dinner, go to sleep, get ready for the next day's festivities. And we'll be talking during the week and next Monday on the podcast, okay?
Yeah, be really good.
Thank you.
You're welcome.
Bye-bye now.
Bye-bye.
In our final session, Arun, Sanyal, and David Kleiner join Roger to explore some prospective benefits of AI-assisted histopathology over individual ordinal reads and discuss an upcoming clinical trial on this topic.
So now we're back for today's extra-sode. As promised, Jörn has stuck around with us, and the others have left and have been replaced by two fantastically esteemed and renowned colleagues. Dr. Arun Sanyal, who's been with us before, in fact, was part of what I think Arun was one of our 3 or 4 most highly rated episodes ever, or highly listened to episodes ever. How are you doing today?
Hey, Roger. It's great to be with you guys and looking forward to a vibrant discussion about AI and pathology.
As are we all. And thank you, Arun. And then our other guest, who's not been on the podcast before, but David, I can't remember how many times I've seen you present. It's just the first time we've ever shared one of these things, is David Kleiner, who is a scientist at the National Cancer Institute. I guess senior research physician is the title. And David, do us a favor, just take a minute and tell our audience, anyone who might not have heard you before, if such a thing exists, a little bit about your background and how you came to be doing what you're doing today.
Well, I've been at the National Cancer Institute for about 35 years now, did my residency here and had an interest in liver disease even before I arrived here, even as a medical student. And so when the opportunities arose to work with the liver disease section here, I grabbed them. And most of my work has been in chronic liver disease. So first with viral hepatitis and primary biliary cholangitis, and more recently with fatty liver disease and drug-induced liver injury. And most of that work has been either in the context of natural history or clinical trials of some sort or another in all of these things. So I guess my expertise is in the evaluation of liver biopsies for the use in databases and clinical trial situations. I've also done some work in liver cancer, but not nearly as much as in chronic liver disease.
David, that's fantastic and really helpful. And I'm going to ask a completely digressive question, which just popped into my head as I listened to you, which is, how is doing this work in NASH and DILI different than it is in the things that you worked on before? Is there a difference, or is it just kind of same thing, different disease?
Yes and no. Chronic viral hepatitis is actually a very simple disease histologically. It's mainly inflammation and fibrosis and the quantification of those two things. Whereas NASH adds in other features that are important, like steatosis and ballooning. So it's a more multidimensional kind of disease, and drug-induced liver injury is all over. So it incorporates vascular injury and biliary injury. And so the system that I developed for evaluating those cases is much more complex. I think one of the nice things about studying viral hepatitis C to begin with was the fact that it is a very stable disease. So from one biopsy, if you haven't treated the patient in between, from one biopsy to the next, the inflammation stays about the same, but the fibrosis will gradually increase. So it's a very well-behaved system, unlike NASH, which tends to go in and out of disease activity, and you may even see fibrosis regression without actually treating the patient. So
that's actually a great jumping off point for the rest of this conversation. Sometimes you ask a question, you get exactly the answer you didn't even think you'd be lucky enough to get. So what we're going to be talking about today is previewing, if you will, a study that's currently ongoing that I'm hoping will be back in several months after data has been published to talk about the results of. But the challenge really is to take a look at how and the degree to which AI-assisted pathology or an interpretation can remedy or ameliorate some of the challenges that we see with kind of traditional ordinal histopathology. And where I think I'd like to start, David, is exactly where you were, and maybe even for you to start, which is today, people point to two sets of challenges around the technology and challenges around reader reliability, both of them having to do with the ability to capture complexity. So if you want to take one of those and dive in on where you see the challenges being today, I'll then ask Arun to handle the other one, and Jörn will ask questions as we go along.
So they are tied together. The technical aspects of reading liver biopsies are tied up in, what happens to the liver biopsy before I even get it. So how big is it? How good was the staining? How good was the sectioning? And when you go to AI, you add another layer on top of that, which is the digital scanning of the slide, which, is subject to its own potential pitfalls that can be addressed. The bugaboo, if you will, of this space has been reader reliability. And there have been obviously lots of studies on reader reliability, some good, some not so good. And I think just to draw the bottom line, is that good reliability is achievable, but that doesn't seem to be the norm. And so for a number of clinical trials, for whatever reason, different pathologists were reading things differently, and that increases the noise of reading. And when you have a situation where the distinction between the before and the after is not great. So just to jump back to the viral hepatitis example, when you treat somebody with an effective antiviral, we don't use histology necessarily to measure the response, but in situations where you did and you chased the virus out of the patient with medication, the activity really goes to very low. The inflammation basically goes away. And that has not been typical in at least the clinical trials that I have seen. If the treatments over the period of time that people were doing these clinical trials really zeroed out the activity, you wouldn't need a scoring system. You could just compare the biopsies next to each other and there would be a dramatic difference. But that has not been the case for one reason or another. And so when we're trying to figure out whether a drug is effective or not, we need measuring tools which are maybe not so noisy as a human is.
So Arun, I noticed you nodding at the end and then furrowing your brow a bit at one point. What about David's comment struck you as being dead on, and what would you like to elaborate on or shift a couple of degrees to the left or right, as it were?
Yeah, first of all, I do not have the temerity to disagree with David. First of all, he's the NASH CRN's chief pathologist, so I can't disagree with him too much because he controls the pathology output from there. No, jokes apart. I think David is right on. As a clinician, we trained in an era where reading histology was part of our clinical training. What we learned the hard way is that there are three sets of things that impact the quality of the histology readout. Things that you do before the sample even arrives in the laboratory, the size of the needle, the gauge of the needle, the kind of quality of the core that you obtain, whether it's fragmented or not, and how long the sample sits on a piece of gauze before somebody decides to put it in formaldehyde and ship it to the lab. All of those kind of things matter. Number two, in the lab, how they process the sample, embed it, and then how the sectioning is done, whether you're getting sections from the juicy middle part of the section or you're just getting the top layer or the bottom layer. All of those things matter. And then sometimes you have a little nick on the blade, which leaves a deep furrow. You have the Grand Canyon going through the middle of the biopsy. That makes it a little more challenging. So all of those things. And then there is the pathologist who then has to interpret all of this, sometimes without any clinical data. We are looking for specificity where many of the histologic findings are not necessarily specific to a given disease state. So all of those are sort of a priori challenges. And I think where the role of AI comes in is to, AI cannot fix, if you put really scraps of tissue on the slide, AI cannot fix that. But assuming you have a reasonable piece of tissue on the slide, then the AI can help the pathologist gain confidence and reduce some of the variability in the histology reading. That's one application. The second application is to learn new things about the histologic evolution of the disease, which is not apparent with conventional staining. And number three is to be able to layer in molecular and other tools to really understand what's happening at a molecular basis so that eventually one day we can theoretically at least match what is going on biologically in the patient to the right drug target, as opposed to throwing the drugs at a bunch of patients hoping for some of them this will be effective.
Something that resounded with me was David commented on the changes in Hep C and how they're not being as prominent in NASH compared to that. And I think, from the learning the field has taken, it was a natural decision to look at changes in liver histology and be able to decide on the outcome of the patient. But it took us some time to understand, from my perspective, that hasn't been as long in the field as Arun or David. That's one of the big learnings, and that we need augmentations of the reading, either by adding blood-based biomarkers, imaging, or, even augmenting the liver histology to actually link this to an outcome. And I think Arun also mentioned something here where he said that there is additional information in the biopsy that might not be described by the NASH CRN NAS score, which it wasn't designed for. And I think at that basis, AI will help us to figure out some of the details in more depth.
So you are on the last one of these that we did, that was sponsored by HistoIndex, was the one that you chaired a few months ago, where we talked about second generation harmonic convergence and the ability ability to compare different drugs to see where in the liver they were having effect. I assume that's the kind of thing that we're talking about when we say learning things we couldn't learn from biopsy.
Well, I'm very intrigued by the type of fibrosis patterns, and David can speak to that in more length. And I think Arun also has seen a lot of that data in the trials he runs. But, fibrosis doesn't always just go away. It starts to kind of dissolve at one point, and I think that's important where you look. And maybe, David, tell me about it, but the current fibrosis staging systems might not be the right ones to study regeneration as much. Is that correct?
So the challenge with fibrosis regression is that it doesn't always look the same as what you would get if the disease progressed from beginning to end in a monotonic way, right? Which is what Hep C does. I keep coming back to that, but it is a really well-behaved system. So what you see in fibrosis regression is, yes, you get two things. You get the collagen becomes more compact, it's basically like sort of an open basket weave going to a very collapsed kind of compact layer. So that's one thing that happens. The fibrosis gets thinner. So what we see as fibrosis under the microscope may be, say, as wide as a hepatocyte, which doesn't necessarily mean anything to your office or your audience, but it's about 50 microns wide, so 1/20th of a millimeter. And it gradually narrows, or it becomes discontinuous. So you get little chunks of collagen that are just sort of sitting there in the middle of the parenchyma. And the scoring systems really don't deal with that very well when you get these disconnected pieces. And so it becomes a challenge just to sort of assign those semi-quantitative numbers to the case. Even so, as a pathologist, I can say, gee, this looks like fibrosis regression. It's not captured by a numbered system. It's just a qualitative observation. But I see that on patients who are just within their natural history. As the activity waxes and wanes, the fibrosis can regress. And I think that kind of throws a wrench into some of the systems sometimes. What we need to do with the AI is learn how to describe those things and measure them in a way that is clinically meaningful, which I think there's still a lot of work to do on that, but it's at least doable.
So first of all, before I turn to Arun, for whom I have a question, I want to note to the audience that every time David mentions the word hepatitis, visualize a thought bubble going up above his head that says, life used to be so simple, because that would capture the look on his face in the moments that he says that. But it's not so simple anymore. So Arun, you brought AI into the conversation in terms of a couple of things that it can do. Could you elaborate a little bit on where you think its greatest benefits are, first of all, in drug development, and then second, in just basic hepatic knowledge?
Well, I think in drug development, probably where the low-hanging fruit is how AI can assist the pathologist. Remember, I'm using the word assist, not replace, but assist the pathologist in having greater confidence in ascertaining the degree of activity and the fibrosis stage. Between the two, there's a tremendous amount of emphasis on fibrosis because fibrosis is what determines prognosis. And so changes in fibrosis are considered highly relevant in terms of translation into what is considered clinically meaningful from a prognostic and a clinical outcomes perspective. And so when the histologic readout for fibrosis is noisy, then you need larger sample sizes and you need bigger effect sizes. So the two consequences of the current background subjective variability in the readouts is that because of this background noise, you need larger sample sizes, and which means bigger studies, and you need bigger effect sizes. So drugs that are not massively effective in wiping out, either activity or fibrosis and are closer to the upper bound of the noise, they have a harder time separating themselves from the noise. Therein lies the challenge. So you could argue if only we had drugs that had more effects on the activity or more effects on the fibrosis, this would not be an issue. But conversely, you could also argue that if you improve the fidelity with which you get a histologic readout, then we would be able to capture things with greater sensitivity. The other point over here is that the fibrosis scoring system is currently an ordinal scoring system. So you go from 1 to 2 to 3 to 4, and the data on prognosis is linked to this ordinal system. But in our early stage studies, where we're trying to figure out whether there's enough action to move forward and do a very large study, you asked about drug development, we may want to capture changes in fibrosis at a more granular and with greater sensitivity. So you have to increase the gain on the readout of the changes in fibrosis. And I think that is where AI tools can really help the pathologist figure out if things are moving or shifting in the right direction, with the caveat caveat that I think both Jörn and David pointed out, that as we published with the tropifexor study, that while fibrosis progresses from stage 1 to 2 to 3 to 4, with drug therapy, especially drug therapy that is targeting the hepatocytes, one of the first places where the collagen cleans up is actually that perihepatic, in the sinusoids, the fibrosis. And so it may take much longer for the actual septa to thin out and break up and other things. So greater certainty in readout of the pathology and being able to identify changes in activity and fibrosis with greater sensitivity. Those are sort of the low-hanging fruit, in my view.
Okay, so one more question to you and then to Jörn. You mentioned ordinal scoring, and then you mentioned the need to be more sensitive. Is ordinal scoring also part of the problem here? Or is that something we can overcome if we simply had more consistent reads or clearer definition from drugs?
Well, I think we have to develop and validate a more, a continuous scale for evaluating the fibrosis. So remember that fibrosis is changing on multiple dimensions. So one is the fibrosis in the sinusoid. One is and within the lobule, there's fibrosis around the central vein. There's fibrosis in the periportal area, and there's fibrosis in the middle. Then you have portal fibrosis, then you have septal fibrosis. So where one of the things we promoted and pushed forward through some of these clinical trial-related studies we did is to look at fibrosis along 5 different axes and summarize it in the form of a radar plot where you can actually see, in which dimension whether the fibrosis is changing and whether it's decreasing, increasing, or remaining stable along each of these different axes. So that gives you a more holistic view of how fibrosis is changing in different parts of the liver section. But I'll let David comment also on if he thinks there's value in some of these things.
I do actually. I think the huge promise of AI is to be able to measure things that are challenging for us to measure. And that's not just the basic scoring that we do, but a lot of other things which may turn out to be very important. And obviously there's a lot of work to be done to show that a particular observation is important. But, Arun touched on fibrosis. You could make the same kind of analogy with all of the other features in the biopsy, where the inflammation is in the portal areas or in the parenchyma, how much there is, how many different foci, how many lymphocytes are actually present in these different areas. With steatosis, you could look at numbers of vacuoles, the size of each individual vacuole, the range of vacuole sizes across the biopsy. I mean, you can really get quite granular with a good AI system. The issue is right now, we don't really know what of those features is important. And that includes the fibrosis. We see these changes, and what we need to do now in clinical trials and other studies is to try and understand what the meaning is of these things. Because if it turns out that the disappearance of perisinusoidal fibrosis, and if that's the first thing that goes away when you treat somebody, if that's a really important clue and it's clinically meaningful, that's a really important piece of information that everybody can use. And we don't have to worry about the more established structural collagen if what we want to do is find out which drug is showing promise early on. But we need to do those studies, and we need to show that that's a meaningful endpoint, because right now all of the studies are based on these scores that humans do, and so everybody is trying to replicate those first.
I think to add on to that, and Arun mentioned the tropifexor study, which has a detailed histopathological AI-supported assessment, is actually, it might be more complex that not every MOA does resolve fibrosis in the same way. Now, I'm just saying this as a hypothesis, but we need more data, and for that reason, I think we need to run these technologies in the trials to also understand it and form the field and know where we're moving. And David said to link this to a clinically meaningful change, it will need some time, but we have the opportunity now, I think.
So moving forward to the third part of the discussion. So Arun, with all that as a background, the study that we're previewing, because we have no data and we're not talking about results, just kind of why and what's in it. What is the goal of the exercise specifically in the context of what we've been talking about so far?
I think the goal of the study is primarily to validate in as rigorous a manner as possible the ability, and this is really focused on fibrosis. It could be, you could design a study to look at other aspects as well, but the focus of this particular study is primarily fibrosis for the reasons I alluded to earlier. And to be able to see whether provision of either second harmonic generated images. So just the image, not any scores or anything, just the image, along with the conventional H&E and serious red staining. Whether provision of that second harmonic generated image gives the pathologist what he needs to be more sure of, what they're looking at. Number two, in addition to these, if you actually provide specific scores, like the quantitative fibrosis score, etc., or the mean septal width and things like that, whether that actually gives the pathologist even more confidence. And of course, it's not just a matter of confidence, it's also a matter of trying to understand whether you can reduce the variance from one pathologist to the other in terms of being able to identify more tightly what a given stage of fibrosis. And lastly, to try and start getting an understanding of, three pathologists could all agree with each other and all three could be wrong. So in the end, we have to also understand how does this relate to ground truth. And I think this study tries to do this to the extent possible, knowing that the only way you can get ground truth is to really take the liver out and section it, 4 microns at a time, which is obviously not feasible. So to the best extent possible to get a handle on the ground truth, but also really to answer whether just provision of some additional, it's like adding another stain. The second harmonic is like, you went from Masson trichrome to serious red, now you got a second harmonic stain, and does that help you or not? Really to answer that question with or without additional actually numerically derived scores, numerical scores to go with it.
David, anything you want to add to that before we dive into exactly what the study specs and parameters are?
So, not to focus on any particular tool because I'm not supposed to do that, but one of the first advantages of using these AI tools is their ability to present a consistent interface to the pathologist, and one that can be evaluated at relatively low magnification. The easier it is to see the changes at, across the whole biopsy at low magnification, the more consistent your scoring will be, your more consistent recognition of features. So whether you're highlighting fibrosis using 2D harmonic generation or some other methodology, or you're highlighting steatosis or ballooning or something else using other methodologies, if you can present that consistent interface to the pathologist, I think just the act of doing that will get you better agreement. And then, beyond that, if the interface is also offering, well, this is where this biopsy falls on the range of 0 to 4, that also is very helpful, because especially when you're not, some things are very clear and some things are not. And so when you're not certain about something, then having a numerical estimate for whatever you're trying to score is helpful.
Okay, that makes sense. Although the only thing I'm wondering is if there's any magic to 0 to 4 in the scheme of things.
So humans are good at differentiating multiple categories up to about 7. And you see that, so there is one staging system, the Ishak staging system, which is 0 to 6, and so 7 categories, and that's reasonably reproducible. But to get any finer than that is difficult. And so we picked 4 just because most staging systems are 0 to 4, and the lines that we were drawing between different stages are comparable to other staging systems that people were familiar with. So we hope to benefit from that. And that was the other scales that we came up with were also ones where we thought we had a group of pathologists for the NASH CRN, and we said, what do you think you can do? How fine a distinction can you make? So, for ballooning, it came out 0, 1, 2. And for other things, it was 0 to 3. So depending on how you look at things, you can get finer distinctions than that. In the just using, again, human observations, we've been able to extend the ballooning scale to 4, 0 to 4, with specific criteria. And that's also reasonably reproducible in our hands.
So, Arun, do us a favor in the few minutes we've got left. Start by walking us through what this design is and how it is designed to address the various issues we've been talking about in this podcast.
Yeah, so the design really involves, first of all, a selection of both experienced and relatively early career pathologists. That gives us an opportunity to look whether the experience of the pathologist makes a difference. Are there based on how much experience do you have, does it make any difference whether AI is more useful in one group versus another? Number 2, we go through a couple of randomizations where a group of biopsy slides that actually cover the spectrum of NASH NASH fibrosis, so from 0 to 4. They are in random order, reviewed by the pathologist using the conventional approach versus having the second harmonic generated image available to them. And this is done in a random order so that every pathologist reviews every biopsy, but none of them review it in the same order. Secondly, in order to reduce a potential bias of pathologists talking to each other about biopsies, etc., or knowing what a more senior pathologist thought it was, the pathologists are actually blinded to each other. So no pathologist participating in the study actually knows who the other pathologists are. So they have been all kept completely individually separated and firewalled, if you will. And then after the first set of readout, then there is a washout period, and then the thing is done all over again. And with the addition of the fibrosis score, the Q fibrosis score in addition to the fibrosis images. And then it is sort of reanalyzed again. And so basically what this does is it allows us to be able to look at how each person scored each biopsy themselves with and without second harmonic generated images in a very random order, and then pairwise comparisons of the pathologists, and then the overall degree of variance with an individual and across the group as a whole, and also to be able to see whether incrementally addition of these numerical scores provides value beyond looking at the image. We also ask the pathologists after each reading to tell us, did they find it useful or not? And then, of course, we can correlate the overall performance with the pathologist's own reading of their own performance and confidence to see whether things did improve or not. So I think that's sort of at a high level how the study is broadly structured. David, did you want to add anything to that?
No, I think you covered it pretty well. The blinding of everybody to everything, multiple observations over a period of time, with and without the assistance of the AI tool. I mean, that's the way that, and with this little survey afterwards, that's the way that this study was designed. And so the idea is that hopefully the AI tool makes things better. What I would emphasize is since none of these pathologists know each other, or at least didn't talk to each other about anything, they didn't even establish their own internal guidelines, right? So there are all kinds of situations that come up when you're looking at fibrosis, and it's important to know, if you're trying to be reproducible with each other, to know how somebody else might interpret a particular situation that comes up. We talk about these stages as if they were very simple things, but every single biopsy you have to make decisions about what you see, and having clear criteria or clear understanding of what everyone is agreeing on when you hit these complicated situations will also increase reproducibility. So they didn't have that. So they started from nothing. So if you see better reproducibility, then that's great, because that's the way most clinical trials are designed, is the pathologists don't talk to each other beforehand.
No, not really. I think this is a very important aspect and a thoroughly thought about scientific, approach in trying to decipher what this type of histological reading, how it can be refined. So no, again, congratulations both, and looking forward to trial results, I guess.
I completely agree. A research scientist I know once told me that anytime you can get the words elegant and rigorous into the same description, you've done something special. And I think both those apply here. So the only other question I've got is, Arun, you made a very, very tantalizing comment about ground truth. Is it reasonable to expect or reasonable to hope that something about ground truth will emerge from this discussion? And if so, what form do you think that'll take?
Well, I think the ground truth eventually will require validation, not only, as we develop more longitudinal studies, we will be able to see whether these more short-term changes translate into more disease or less disease in the long term. That's one way to sort of get to the ground truth. Another way is to look at a lot of NITs and see, the concordance. If 5 different NITs also agree with the direction of the reading of the pathologist, that's another way to sort of think about ground truth. And lastly, of course, ground truth also has to look at collagen quantification, some of these other type of more quantitative tools that we can see if the pathologist all said the fibrosis was worsening and the proportionate area increased. That's another sort of way of sort of getting that, yes, things were getting worse. Or conversely, things are getting better. So I think there are different ways, because this comes up all the time when we're talking about validation of some of these AI tools, particularly with regulatory agencies. But it is not easy to establish ground truth when the liver is still sitting inside the patient, and none of the NITs have been validated either to represent the ground truth. So the best, it's like infinity, you can approach infinity, you cannot actually be at infinity. So I think ground truth is an aspirational goal. But through all of these, I think this will certainly generate, I think the results of the study will generate a very robust conversation and hopefully some innovation about some best practices in trying to establish the ground truths. Because in the end, there is a huge impact when we make a false assumption and either kill a drug that actually is potentially beneficial, or we bring a drug forward into phase 3 and then fail miserably. And there are probably instances certainly of drugs being moved into phase 3 and failing in phase 3 in the NASH space that we all remember. So the closer we can get to this important question, the better it'll be for the field and for all our patients.
Yeah, and to just elaborate on what you said, given my background as a forecaster, quick, correct kills in development are the single most cost-effective thing a manufacturer can do. So amen to that. Look, this has been a fantastic conversation. I'm excited to see the results of the study. Jörn, David, Arun, thanks for a fantastic discussion, and I certainly emerged feeling a lot better educated. I think our listeners will as well. And look forward to seeing you guys back in November when we can talk about the actual, or at some point in the future, we can actually talk about the results of this thing. Everyone have a good afternoon.
Thank you. Great seeing you.
Thank you. Bye, guys.
Bye-bye now.
As Roger said earlier, there will be no business section today.
We will release 5 conversations over the weekend, but no vault.
Episode 28, our preview of the ADA meeting in San Diego, will post next Wednesday. Today's episode has been sponsored by HistoIndex, the first global company to provide stain-free, fully automated imaging solutions for visualizing and quantifying morphological and architectural features of fibrosis.
HistoIndex couples breakthrough biophotonic technology with an AI-powered digital pathology system to provide consistent, high-throughput imaging in liver disease and cancer.
The platform benefits clinical research and speeds development of drugs and diagnostics.
For more information, visit www.histoindex.com.
Have any questions for the surfers? You can send them to surfingnash.com, and we will answer on the podcast or the website.